So welcome to the first of the live session of the Iso-Seq Social Club, Volume 3. So let's get started with our first speaker of the day. Denis, if you can come on stage, please. So go ahead.
Okay. Thank you very much, Liz, for the introduction, and good morning, everybody. My name is Denis Torre. I am a PhD student in the labs of Robert Sebra and Ernesto Guccione at the Icahn School of Medicine at Mount Sinai in New York. And today I'm going to highlight some of the more recent results for one of our ongoing projects that involves applying PacBio Iso-Seq to studying the transcriptome of early human embryos. Okay. Human preimplantation development is a highly complex process during which the transcriptome is extensively remodeled. Early embryos inherit large amounts of RNA from the oocyte, which is known as maternal mRNA, and then subsequently start transcribing their own mRNA through several processes or phases known as embryonic genome activation, or EGA, the major of which takes place between the transition from the 4-cell to the 8-cell stages. So overall, one of the key questions in mammalian and human embryology is understanding how these vast changes in gene expression are orchestrated to meet the needs of the developing embryo. And so far, several multiomics data sets have been performed upon samples from these stages, both in the transcriptomics and in the genetic space. However, so far, there are no isoform-resolved RNA-seq data sets from human preimplantation stages yet. So the goal of this study is to profile the early human transcriptome by using a combined long and short-read RNA sequencing approach. The embryos used in this study were obtained through our collaborator Dalit Ben Yosef, who is the director of the IVF unit at the Tel Aviv Sourasky Medical Center. So the embryos used in this study were produced by in vitro fertilization for clinical purposes. And then surplus embryos that were not used for fertility purposes were frozen and donated for research with full informed consent by the parents. We had access to a total of 73 embryos between the zygote and the blastocyst stages, which underwent RNA extraction followed by a combined long and short-read RNA sequencing. The resulting data was then integrated using a computational pipeline that leverages the Squanti tool, which was discussed earlier today in Francisco's great talk. And specifically, we used the long-read data to define and identify novel isoforms and classify them based on their similarity to known isoforms. And then we leveraged the short-read RNA-seq data to validate the novel splice junctions identified in the transcriptome, and then quantify both expression and splicing throughout development. We then leveraged the novel transcriptome to perform a series of downstream analysis, which I'm going to discuss in the next few slides. So here I'm showing a summary table of the classification of the isoforms that we used to define the transcripts and produce the final reference set. Overall, in addition to known isoforms, we included novel in catalog and novel not in catalog, which are two different classes of novel isoforms of known genes that specifically contain novel combination of known splice sites, or at least one novel splice site. And then novel antisense or heterogenic isoforms, which are transcribed from novel genes that are not present in the currently available reference transcriptome. For this specific study, we made the decision to remove any novel monoexonic and incomplete splice matches from the reference in order to produce the most high quality and full-length transcriptome possible that is also highly compatible with downstream Illumina quantification from whomever might be interested in using this resource. Overall, we identified over 100,000 novel isoforms, the largest class being the novel not in catalog and followed by the novel in catalog categories. We also found over 9,000 novel antisense isoforms and over 8,000 novel intergenic isoforms, which are collectively transcribed by about 5,200 novel unannotated genes that are not present in the latest release of the Ensemble Human Transcriptome. We then applied multiple computational tools and integrated multiple publicly available data sets to further characterize and validate the isoforms in the transcriptome. In terms of computational tools, we predicted the protein coding probability, protein domain content, repetitive element content, and evolutionary conservation of the isoforms. And in parallel, we also integrated both short-read RNA-seq datasets and several epigenetic datasets, such as ATAC-seq, cutting run for promoter marks, and DNA methylation, to further validate the isoform expression and the marks surrounding their transcriptional start sites. Here I'm showing some of the results from the protein coding potential analysis. So to perform this analysis, we took the nucleotide sequence of all the full-length isoforms in our transcriptome and then applied the protein coding prediction tool CPAT to calculate the protein coding probability for each of these isoforms. For ones that were predicted to be protein coding, we then extracted the best open reading frame sequence, translated it to amino acid sequence, and then used PFAM scan to identify whether any of these predicted peptides contain known protein domains. So on the right, I'm showing the results of this analysis, where we can see that the majority of isoforms transcribed from known genes, both known, novel in catalog, and novel not in catalog, are predicted to produce a protein that contains at least one known domain. By contrast, instead, novel antisense and intergenic isoforms are overwhelmingly predicted to be non-coding, thus suggesting that early human embryos may transcribe large amounts of previously uncharacterized non-coding RNAs. Next, we further characterize these isoforms by scanning their nucleotide sequence for the integration of repetitive elements that are commonly dispersed throughout the genome. These include lines, signs, and transposable elements such as LTRs. We specifically divided these integration events into 3 different classes based on whether the repetitive element is included at the promoter of the isoform, internally, or at the transcriptional end site of the isoform. Some of the highlights of this analysis are the fact that over a quarter of novel antisense and intergenic isoforms appear to be driven by an LTR promoter or repetitive promoter. And some of these include the human endogenous retrovirus HUVE, which is known to be highly activated in early stages of development. We also find widespread evidence of internal repeat inclusion. This is especially evident in novel non-catalog isoforms for known genes. And again, novel antisense and intergenic isoforms are specifically repeat-rich. We also find some evidence of repeat-derived terminators, though less so. And again, this seems to be primarily concentrated among antisense and intergenic isoforms. Next, we also sought to profile the evolutionary conservation of isoforms at the species level by performing a blast analysis, so scanning the isoform nucleotide sequence on the nucleotide sequence of several reference genomes from vertebrates of varying degrees of similarity from the human. And we specifically defined a hit between an isoform and a genome based on the presence of an alignment of at least 100 base pairs with at least 95% sequence identity. So predictably, we see very high levels of similarity between these isoforms and the genomes of very close primates to the human, such as the chimp and the gorilla. But some of the more striking results are found in the antisense and intergenic class, which appear to drop off very rapidly in conservation. And by the time we get to prosimians and even further away species such as rodents, we have close to 0% conservation. By contrast, instead, the different classes of isoforms transcribed from known genes appear to be more conserved even in farther away species. So these results suggest that some of these novel genes we found in early human embryos may be products of more recent evolutionary events that set hominids and humans aside from other mammals. Next, as I mentioned before, we also integrated both our own short-read RNA-seq data and 3 publicly available short-read RNA-seq datasets that analyzed human embryos in comparable stages. On the x-axis, I'm showing the percentage of isoforms for each class that are either not expressed, lowly expressed, or highly expressed at each of the developmental stages listed in all these different datasets. As you can see, the classes of all isoform classes, both known and novel, seem to be widely expressed across both datasets and studies and developmental stages. And again, some of the more interesting results lie with the novel antisense and intergenic isoforms that appear to reach highest levels of expression between the 4 to 8 cell stages in development, both in our data and other datasets, and then are subsequently downregulated and reach our lowest levels in the blastocyst. So together, these data suggest that the current reference transcriptome, in this case, we use the ensemble reference, appears to lack some of these novel isoforms that are expressed in early embryos. And this might be to historical limitations of short-read RNA-seq data, which were unable to reconstruct the full length of these isoforms, potentially especially for repeat-rich RNAs, and also due to the difficulty in sample procurement of these early stage samples. Next, we also start to profile the co-expression dynamics of both known and novel genes throughout development using a non-biased approach. So we applied gene-gene co-expression network analysis, or WGCNA, on a matrix of gene expression levels that was quantified using our SCM across our 73 human embryos between the zygote and the blastocyst stages. On the left, I'm showing a hierarchical clustering dendrogram of our data, which also highlights the modules to which all the genes were assigned, together with normalized expression levels between the one cell to the blastocyst stages. And we also highlight the presence of novel genes within this clustering dendrogram with these black bars shown on the second row of the dendrogram. You can very clearly see that novel genes tend to cluster primarily with early expressed genes that are highly expressed between the 1 to 8 cell stages. And some of the more interesting results from this analysis was identifying a couple of small modules that appeared to be very transiently expressed, either at the 4 cell or the 8 cell stage, and then downregulated, which are very significantly enriched with novel genes, and they may therefore represent yet uncharacterized modules of both protein coding and non-coding elements that may play a role in embryonic genome activation. On the right-hand side, I'm also showing a network diagram of the different modules and highlighting some of the key developmental regulators in embryonic development, including SOX2, NANOG, and MYC. Lastly, as I move to wrap up the talk, I'm just going to showcase a couple examples of novel isoforms of known regulators in embryonic development. On the left, I'm showing short-read RNA-seq pileup across stages together with long-read isoforms of DNMT1, DNA methyltransferase, which is maternally inherited and plays a role in the maintenance of DNA imprints. Specifically, we find evidence for novel transcriptional start site, which appears to be the major TSS used in early stages. We also find novel in-frame exons, novel transcriptional end sites, and a novel short isoform, which appears to be highly used at the 8 cell stage. And all these isoforms contain different combination of orth lengths and predicted protein domains, so may represent potential aspects of DNMT1 biology that are still uncharacterized. A more simple example is shown on the right, where we have a couple new isoforms of FOXO3, which is a fork-head-boss transcription factor. And specifically, we highlight novel transcriptional start site, as well as novel in-frame exons, all of which, again, are predicted to affect the open reading frame sequence, and thus potentially the protein product and its downstream effector functions. So lastly, moving on to the conclusions, as a summary, we applied an integrated PacBio Iso-Seq and short-read RNA-seq approach to profile the transcriptomes of 73 human embryos across stages from the zygote to the blastocyst. And overall, this allowed us to identify over 100,000 novel isoforms that display significant differences in predicted properties such as protein coding potential, inclusion of repetitive elements, and evolutionary conservation. We then integrated multiple published data sets and performed a co-expression network analysis, further showing that most novel isoforms in genes are expressed in early stages and are subsequently downregulated. That being said, I'd like to thank you all for the attention. Thanks a lot for Liz for inviting me to give this talk. Specifically want to thank my supervisors, Robert Sabra and Nessa Guccione, all the other lab members, and specifically Nancy Francoeur, who is a co-lead author on this project and helped set this up, as well as our collaborator Dilit, without whom we would not have had access to the embryo samples and the project would have not been possible. Thanks again all for the attention, and I'm happy to take any questions if there's time.
So Denis, there are amazing questions in the Q&A chat. In fact, they're very complicated, so I hope that you can take some time to read it and maybe respond through the text, or we can do it. We can actually answer some of these questions after the meeting as well. In the interest of time, I'm going to ask Sydney, our next speaker, to come on stage.
Hi, everyone. Can you hear me okay?
Yes, take it away.
All right, sounds good. Hi, everyone. My name's Sydney. I am a grad student at Yale University, and I'm going to be talking to you today about a project that very much work in progress, but where we're trying to understand the boundaries between PTSD and depression through studying alternative splicing. Okay, so PTSD is a super heterogeneous neuropsychiatric disorder and is characterized by a wide variety of different symptoms. In fact, according to the DSM-5, there are over 600,000 combinations of symptoms that can qualify someone for a PTSD diagnosis. And partially because of this, it's very clinically complicated for us to treat. So currently, exposure-based cognitive behavioral therapy is the best sort of course of action. And there are a couple of antidepressant medications that are FDA approved for treatment of PTSD. But basically, there's a huge need to better understand the molecular pathways involved in this condition. In order to potentially facilitate better understanding and better treatment of it. Previous studies that have explored the transcriptome in PTSD have identified large-scale changes both at the gene and exon level. And there's also been a lot of interest in comparing between psychiatric disorders. So both at the level of genetic variation and also transcriptomics, like gene expression, we see striking overlap between many psychiatric disorders. It's shown in the figures here. And in the bottom one, you can see specifically between major depressive disorder, or depression, or MDD, and PTSD, we see like a high, but maybe not quite significant in this study, correlation at the transcriptomic level. However, MDD and PTSD are very clearly distinct disorders with separate etiologies and sometimes different symptoms. And so the question that we're trying to get at is if these disorders have such similar genetic risk and similar transcriptomic profiles, what can we find at the molecular level that actually distinguishes them? And so to begin to delve into this, we started with a large postmortem data set from human brain tissue. We have short-read RNA sequencing from 4 brain regions, 2 regions of the cortex and 2 regions of the amygdala. And as you can see in the table here, we have over 100 sequenced samples from each of those 4 brain regions in control, MDD, and PTSD subjects. So in this forum, I don't think I necessarily need to explain what alternative splicing is or why we're so interested in it, but I'll just mention that more and more alternative splicing has been implicated in disease as a potentially really crucial disease mechanism. And isoform-level dysregulation has been previously studied in various other psychiatric or neurodevelopmental disorders, but not previously in PTSD. So I began this analysis using a software called LeafCutter to analyze differential splicing events. I ran this analysis using the 7 covariates that are listed here. And in this table, you can see the numbers of of genes with at least one differential splicing event that we identify in PTSD versus control and MDD versus control in each of our 4 brain regions. So let's see. Okay. Given that we're interested in comparing between these regions and samples, this is an upset plot showing the overlap between the genes that show differential splicing in each comparison. And hopefully what you can appreciate is that most of the largest sets of genes are in the, sorry, there's a lag here, whatever, are unique to any one condition or region. And so we don't see a whole lot of overlap between them, which indicates that there are unique sets of genes that are differentially spliced in each of these settings. When we did some gene ontology analysis, we're showing here each row represents a specific gene. If it's red, it's present in the list shown at the bottom of the column. And the list names that are in bold are significantly enriched for that category. So for example, you can see that many of our comparisons are significantly enriched for axon guidance genes, but different genes seem to be driving that enrichment in each of our conditions. So basically, we think that different genes are being differentially spliced in each of these regions and in PTSD and MDD, but they seem to be converging on common molecular functions or pathways. The final bit of short read analysis that I want to show you here is an overall correlation in the transcriptomes and the spliceomes. So you can see on the left that these 4 plots correlate the expression values for all of the genes detected in the genome between PTSD and MDD in each of the 4 regions. And the plots on the right show the same thing, but for the splicing patterns of all of the genes. And so hopefully what you can appreciate is that all of the correlations are stronger in the expression plots than in the splicing plots. So basically we're showing that MDD and PTSD are more similar in terms of their expression profiles than their splicing profiles. So this really confirms what we were hoping to see, which is that studying alternative splicing can help us understand what is distinct about MDD and PTSD. Okay, so I also don't think I need to justify in this forum why using long-read sequencing can help to enhance this analysis. So parsing together the short-read information to try to identify information about splicing patterns or the presence of various isoforms gives us some information. But of course, by doing long-read sequencing, we get much more information about the exact isoforms that are present in each of our samples. So we're in the process of collecting a large Iso-Seq data set. We are using the same tissue from those same 4 brain regions from PTSD, MDD, and control samples. We have about 55 samples that have already been sequenced and processed, and that I'm using for the slides that I'm showing you here. But our goal is to have about 7 to 10 samples per category here. So what I'm going to be showing you is some preliminary analysis to compare PTSD and MDD within this Iso-Seq data set. What I've done here is taken the output of SCONTI3, counted instances of specific isoforms within each condition, and then done sort of what I think is the most stringent analysis for unique isoforms. So I'm looking for isoforms that are solely found in the PTSD or MDD samples and not seen at all in the control samples. This is a plot of the structural categories of those isoforms that we're seeing. I think there's some potentially interesting patterns here, but also it might indicate that we could do some more filtering. So I actually haven't yet done that filtering and then recovery process that was talked about in SCONTI3 in the previous session. So I think that might be a good next step here. But we see lots of novel in catalog and novel not in catalog isoforms, and those are also really interesting for us to delve into. In terms of overlap, we see similar pattern to what we see in the differentially spliced genes in the short read analysis, which is that there isn't very much overlap between the different lists. And most of the overlap that you do see is with the lists in the medial amygdala, which you might have noticed seem to have a lot more diversity, both at the splicing and gene expression level than other regions. That's a finding that seems to be replicated across a lot of data sets, and we don't fully understand, but we see that here as well. The final thing I want to show you is that we did a preliminary overlap between our long read and our short read analyses just at the level of genes. So we're looking for genes that have at least one novel isoform in the long read and have at least one differential splicing event in the short read analysis. And we do see some overlap. Once again, a lot in the medial amygdala where the lists are just very large. But some of these genes that we're seeing here have been previously implicated in neuropsychiatric disorders or have functions that make them sort of biologically plausible. So there's a lot of interesting stuff for us to delve into here. So to conclude, we've used this large-scale transcriptomic data set to identify alternative splicing patterns that help us understand the boundaries between PTSD and MDD. We found sets of distinct genes that were differentially spliced in each region and condition, but they seem to converge on common molecular pathways. We see that MDD and PTSD are more distinct in terms of their splicing profiles than their expression profiles. And we're definitely seeing that Iso-Seq can help us identify unique isoforms to MDD and PTSD in each of our regions. The thing that's definitely ongoing and that I'm working on, because I'm also new to this analysis, is how to combine these data types more effectively. So if you all have suggestions or thoughts about ways to compare between conditions within Iso-Seq analysis, or to integrate our Iso-Seq and our short read analysis, not just at sort of the gene level, but at the specific level of like isoforms and junctions. I'd love to hear your suggestions, and I'm hoping some of the other talks today will share ideas about those things. So with that, I want to thank Matt Turgenti, who's the faculty member at Yale that I'm working with this project on, my thesis advisor, Nana Cheston, and everyone who's contributed to this work, and you all for listening. So thank you very much.
Thanks, Sydney. And we have time. There is a question that I'll bring up, and I might have a comment on the question in your slide, in the previous slide.
Cool.
So the question from the audience is, what is the reasoning for using LeafCutter against other splice quantification tools?
That's a great question. I think the honest answer is just that we looked at previous work, and it seemed to be a very commonly used method that was well documented and fit well with the kind of results that we were looking for. So that's why we went with it. If there are suggestions of other algorithms to use or alternatives, we'd love to hear them, but that seemed to be the standard. And so that's what we went with.
And in terms of integrating the data set, sorry, there's another question. I'd like to prioritize the audience question over mine. When you look at the overlap between isoforms for different conditions, are you looking for full overlap, partial overlap, or overlap between exons?
So in the analysis that I'm doing so far, I'm trying to do this in sort of the like strictest way possible. And so what I did was look for like full matches. So like every single junction in an isoform had to be exactly the same across the samples in order to overlap. So that doesn't mean that even small differences would be considered different isoforms, but we tried to be very strict about matching them across samples.
Cool. I think maybe my last comment on integration is that I know there's actually a lot of our speakers and even some of the people in attendance here have actually experienced with integrating a large Iso-Seq data sets with RNA-seq. Angeles' talk on tapas, which is one of the recorded sessions, I think is that. And then Gloria's lab, so Madison is from Gloria's lab that's speaking later. They've also been thinking very hard about integrating large data sets.
Yeah, that's awesome. Just from the titles, I kind of inferred that. And I'm excited to learn from those sessions too. So.
Cool.
Yeah, thank you.
Thank you. Let's have our next speaker, Pratibha.
Okay, good morning, everybody. My name is Pratibha, and today I'll be presenting on a new technology from the Yale lab at UCSD. The technology is long-read stamp, and it's used for mapping RBP-RNA interactions in an isoform-aware manner. So RNA binding proteins are known to be involved in critical stages of RNA life. This can include transcription, splicing, modification, intercellular trafficking, translation, and decay. And it's estimated that there are over 4,000 RNA binding proteins, and that alterations in RNA binding proteins are known to be implicated in disease progression. In terms of gold standard methods for studying RBP-RNA interactions, the current gold standard method includes methods like CLIP. However, these methods have limitations. These include that these methods are labor intensive. They often require transcript fragmentation, which means it's really difficult to study RBP-RNA interactions at the isoform level. And these methods also often require large amounts of input material, which is limiting for certain types of experiments, such as single cell. So our lab is part of the ENCODE consortium, and as a part of that consortium, we've been working really hard to characterize RNA binding proteins using methods like CLIP and other methods listed over here. And we've been able to characterize about 20% of RNA binding proteins. However, many more remain to be characterized. And so we're constantly looking for better methods and developing better methods to do this. And so along kind of that vein, we started asking the question, of can we develop other scalable methods to identify RNA binding protein targets in further detail. So when thinking about this, we developed STAMP, which belongs into this new category of technologies that involves using an RNA binding protein to RNA editing enzymes. And to explain how this works, I'm going to use an analogy. And the analogy is take your kid to work day. So imagine there's this kid, and the kid represents an RNA editing enzyme. And this kid really likes red confetti. And he's going to work with his dad that represents the RNA binding protein. Now, you would assume that on Take Your Kid to Work Day, wherever the dad goes, the kid's going to go as well, and so is the confetti. And so by looking for the presence or the absence of red confetti, we can make inferences about where the dad has or has not been at work. And so this is exactly how STAMP works. And so in STAMP, we use APOBEC1, which is an RNA editing enzyme. And basically, by looking for APOBEC1 edits, these edits can indicate where the RNA binding protein interacts with the RNA. And this is something that we can determine just by looking at standard RNA-seq reads. So in terms of other technologies within this class, another popular method is TRIBE. However, TRIBE uses ADAR, and the limitation of this method is that ADAR, when fused to an RNA binding protein and its just native function, it primarily edits only double-stranded RNA, so we're limited in the information that we can gather. So to address the limitations of current gold standard methods for studying RBP-RNA interactions, as well as current technologies in this new class of technologies that I just talked about, we developed STAMP, which again involves using APOBEC1. We fuse an RNA binding protein to APOBEC1, and we look for C-to-U edits characteristic of APOBEC1 just in standard RNA-seq reads. We use Sailor, which is a quantification pipeline that we've developed in-house. This is a published method. From there, we can determine gene targets and binding sites. So as I mentioned, this method can be coupled with standard sequencing methods, and by that I mean it can be coupled with short-read sequencing, long-read sequencing, and single-cell sequencing. So I'm going to start by talking about short-read sequencing because this is where we did a lot of our proof of concept work. So for our proof of concept, we started by using RBFOX2, which is a really well-established RNA binding protein. We fused it to APOBEC1. In the future, I'll be calling this RBFOX2 STAMP. And we did this experiment using short-read sequencing data. And over here, basically what I'm showing is a known RBFOX2 target, APP. This is specifically the 3' UTR region. In this first lane over here, we're showing RBFOX2 native eClip data, and these peaks indicate that RBFOX2 binds to the 3' UTR region. Now, in this next lane, what I'm showing is control STAMP, which is just basically only APOBEC editing. In these coverage plots, we can see edits that are indicated by these orange lines. And basically, in the control STAMP, we see very minimal editing. In RBFOX2 STAMP, we see quite a bit of editing. And if we just pull out the edits in these next two lanes, again, in control, we see pretty much nothing. In RBFOX2 STAMP, we see quite a bit of editing, and the editing lines up pretty well with these peaks that we see over here in the eClip data. So this was promising to start with. Now, the next thing we wanted to check is whether the edits are reproducible. So STAMP is a DOX-inducible system. So in these three different types of DOX inductions, which is no DOX, low DOX, and high DOX, we were looking for the reproducibility of edits between replicates. So over here, we have replicate 1 on the x-axis, replicate 2 on the y-axis, and then in the low DOX and high DOX, as expected, we see that the edits are pretty reproducible. Moving on to evaluating specificity of STAMP. As I mentioned, RBFOX2 is a really well-established RNA binding protein, and it has a well-established motif that's associated with where it binds, and this motif is GC-AUG. So if STAMP has high specificity, we would expect to find that the edits are near this GC-AUG motif. And following motif enrichment analysis, this is exactly what we saw as expected in the low DOX induction and high DOX induction conditions. So this indicated to us that STAMP does have high specificity. So from here, we moved on to long-read sequencing. Now, why even couple STAMP with long-read sequencing? Well, we know that long-read sequencing is a really great tool in order to be able to study things at the isoform level. And it's important to study RBP-RNA interactions at the isoform level because with isoforms, they're basically transcripts that occur at the same locus but can differ in transcription start sites and transcription end sites and CDS regions. These are all regions where RBPs can interact with the transcript in different ways. So it's important if we're trying to characterize RNA binding proteins and how they interact with their targets to be able to do this at the isoform level. So again, we first used RBFox2 STAMP, and the first thing that we wanted to do was evaluate the two most popular long-read sequencing platforms, which is Oxford Nanopore Technologies and PacBio sequencing. So again, over here, I'm showing APP. This is the 3' UTR region of APP. Our eclipse peaks indicate that RBFox2 does bind to this region. Our short-read sequencing data in the control, we see very minimal editing. In RBFox2 STAMP, we see some editing that lines up pretty well with these peaks as expected. Moving on to the long-read platforms, we saw quite a bit of editing in our control STAMP for Oxford Nanopore Technologies. We did see some editing in the RBFox2 STAMP for the Oxford Nanopore Technologies long-read data. However, it was really hard to separate out kind of the signal from the background noise, and this is most likely due to the higher error rate that's currently associated with this platform. Moving on to PacBio, we see that the control is really clean with pretty much no editing at all, as expected. And in RBFox2 STAMP, we see quite a bit of editing that does line up well with these peaks in the eclipse data. And so just from this data, we gathered that PacBio is probably the way to go, but we also wanted to do a motif enrichment analysis on the PacBio data and the Oxford Nanopore Technology data. And we found that we were able to pull out this GC-AUG motif pretty well with both, but with much higher significance in PacBio. So we decided to continue using the PacBio platform. So one of the advantages of STAMP is we're able to do a transcriptome-wide analysis pretty easily. And so over here, I'm showing the transcriptome-wide analysis for RBFox2 STAMP, and we're able to see differences in RBFox2 STAMP edits between isoforms, and we don't see these same differences in the control sample. So I'm going to highlight a couple of specific examples here that will hopefully motivate the strengths of this technology. So over here, one of the candidates that came up was FAR1. So this is the 3' UTR region of FAR1. Hopefully this format of the figure is familiar to you all by now, but in the first lane, we are seeing eclipse peaks here indicating that RBFox2 binds to this region. Looking at the short read data in the control, we don't see any editing really. In RBFox2 STAMP, we see a fair amount of editing that lines up well with the peaks. Now looking at the kind of all the reads for this gene in the long read. In the control, we've seen no editing for the PacBio long read data. In RBFox2 STAMP, we see quite a bit of editing, but only when we look at the reads associated with these individual isoforms can we understand that the editing is pretty much exclusive to this isoform with a longer 3' UTR, and we see pretty much no editing for this second isoform, which I'm calling isoform B. So over here, I'm showing another example. This is pig N. This is another example where we can see that the editing is pretty much exclusive to this longer 3' UTR, this isoform with the longer 3' UTR, and the shorter, the isoform with the shorter 3' UTR doesn't really have that much editing. So in terms of the computational pipeline, we start with the raw PacBio reads. We run it through the standard Iso-Seq version 3 pipeline. We use a slightly modified version of Sailor, which again is a published method. And from there, we get the edits, and we write custom scripts based on the downstream analysis that we're looking to do. So from here, there's quite a few ways in which we hope to use the technology and expand the technology. So we think that this technology can be used to study isoform-level translation by fusing RPS2, a ribosomal subunit, with APOBEC1. This is something that we've already done in short-read sequencing. We also hope to use this technology to study RBP-RNA interactions in different cell types. And then we also hope to include a second RNA editing enzyme in order to study RBP regulatory interplay. So in this talk, I hope that I've convinced you that STAMP is a new approach for studying RNA-protein interactions, read sequencing, long-read sequencing, and single-cell sequencing, which I didn't get to talk about much in this talk, but we do have more details on it in our paper. And lastly, when coupled with long-read sequencing, STAMP can be used to survey RNA-binding protein targets and interaction sites at isoform-level resolution. So before taking questions, I'd like to acknowledge my PI, Jean Yeo, as well as all of my co-authors on this paper. There's quite a bit that I didn't get to talk about in this talk that is addressed in the paper, so if this sounds interesting to you, please do check it out. Also, a huge shout out to Liz, who helped guide us a lot through the computational analysis for this method. And I'd also like to acknowledge our funding sources. And I will take questions.
All right. So in the interest of time, I'm gonna bring up our last speaker, Madison.
First and foremost, thank you everyone for coming to my talk today. I'm Madison Mehlferber, a PhD candidate within Dr. Gloria Shankman's lab at the University of Virginia. And I'll be talking to you today about my talk entitled Characterization of Protein Isoform Diversity in Human Umbilical Vein Endothelial Cells via Long-Read Proteogenomics. So first and foremost, why are we interested in endothelial cells? Well, endothelial cells are essential cell types within the body. They form the luminal lining of all the blood vessels within our cardiovascular system, allowing for the establishment of critical functions of the cardiovascular system, inclusive of blood pressure regulation, gas exchange, waste removal, anticoagulation, and nutrient delivery. Endothelial cells themselves are heterogeneous cell types, and through complex signaling cascades, they can differentiate and specify into their various subtypes, inclusive of arteries, capillaries, veins, and hemogenic cell types. However, the proteins underlying their specification events that allow for them to take on these different phenotypes remain poorly characterized. A source to which we know that heterogeneity can be achieved within cell populations is through the process of alternative splicing, where we can get this establishment of different mRNA pools, which can produce functionally distinct proteins through different processes. Endothelial cells themselves have been shown to partake within this alternative splicing pathway and produce functionally distinct isoforms. And one of the examples which I want to highlight that's very critical within the cardiovascular realm is within the VEGF-R2 signaling pathway. So the loss of an exon within the VEGF-A receptor gene can cause this establishment of these two separate isoform families, one that promotes the formation of new blood vessels and one isoform family that actually hinders this process. And they work together in balance within the cardiovascular system. So the goal of my research is really to develop a systematic method to enhance characterization of the protein isoform landscape within endothelial cells. If we can pinpoint endothelial-specific isoform expression patterns, and can we understand the contribution of isoforms to the establishment of endothelial cell identity? So I hope I've convinced you that the characterization of isoforms is critical to providing insights into cell phenotype. Therefore, we need a way to measure isoforms in a systematic manner. So I asked the question, are the current sequencing techniques alone enough to elucidate splice networks in a cell-specific context? We know that sequencing resolution is integral for isoform detection. Due to the short fragments that are resulting from short-read RNA sequencing, we can't really get at the identity of isoforms, so it's therefore not really ideal for isoform detection in this context. However, we can turn to PacBio long-read sequencing, where we can have the full-length transcripts within our sample and have the entire identity of the isoform populations within a cell-specific context. We do need to understand if transcript expression is also seen at the protein level, so we need to see what protein isoforms are actually expressed within endothelial cell populations to give protein-level evidence for these different isoform populations. So we can turn to proteomics, where we can take our cells of interest, lace them open to get out their proteins, proteolytically digest them into peptides that become amenable for mass spectra sequencing, and then we can get these mass spectra, which are representations of the actual protein products within our cell populations. So together we can integrate this. We can integrate long-read transcriptomic data with that of proteomics data for what we call long-read proteogenomics. This paper and this method development has already been out within Genobiology. I encourage you to look at this publication if you're interested in further descriptions of this method development. And we've also had a recent shout out within Nature Methods. But I'll take you through my project in which I have demonstrated application of long-read proteogenomics on human umbilical vein endothelial cells for this purpose of protein isoform identification and understand this landscape within endothelial cells. So the long-read proteogenomics platform is really separated into two arms of analysis, as I kind of alluded to earlier. This first being this long-read sequencing arm of analysis, where we're taking our cells of interest, subjecting them to PacBio sequencing, predicting their full-length proteins, and establishing this into a sample-specific database for which we can use for downstream protein evidence in a later stage. I know it's a little complicated, but I will take you through the first step of this pipeline, which is this long-read sequencing aspect. So that's exactly what I did. I performed PacBio long-read sequencing on human umbilical vein endothelial cells, where I was able to identify new and novel isoforms. Because we have the full identity of the transcripts within our sample, we can compare those transcripts against the reference to see for which we're finding known isoforms and for which we're finding novel, where those transcripts are differing from what's already been shown within the reference database. Since the goal of this project is really to discover multiple co-expressing isoforms from a gene, I wanted to look at the distribution for which how many genes are actually expressing multiple isoforms within my population. So here you're looking at a distribution of the genes with abundance thresholds of greater than 1 full-length count per million, demonstrating expression of multiple isoforms per gene. You can see that most genes are expressing one isoform, but there are many genes that are expressing more than one isoform. So since I've kind of just told you about this RNA level of things, this transcript level, I want to take this to the protein level, where we can look at the actual existence of these transcripts at the protein level. So this will bring us to our proteomics arm of the long-read proteogenomics pipeline. So the peptides that we identify via mass spectrometry are the ones that are helping us to identify genes. So these mass spectra that we're generating through taking out the proteins of these cells and enzymatically digesting them become a way for us to search our database to figure out for what proteins do we have expressed within our sample. These spectra are representations of the actual proteins within our sample. So we're basically getting two sets of outputs here. We're getting a list of the peptides that are identified, where we found over 99,000, and a list of the genes with any sort of peptide evidence to give them some sort of level of protein evidence. And we found over 10,000 genes within this category. But I'm asking the question of protein isoforms. So how can we infer the presence of protein isoforms from the peptides that we find? So protein isoform identification is limited by the sequences that are in the database. So if I guide your attention towards the green segments in this image, it's very much like short-read RNA sequencing. I can't discern in the first scenario where that peptide came from, what isoform was contributing that peptide at the protein level. I can't tell you that information. However, if I draw your attention towards the second scenario, where we have the pink segment that matches up to the one isoform, I can definitively say that that peptide was contributed by that one isoform, and I can give protein level evidence to that transcript and that protein isoform, therefore. And we can have multiple situations of this where we're finding more unique isoforms through these unique peptide regions. So basically, we need to find a way for us to get a better database for us to find more situations in which we're finding more concrete protein level evidence. So basically what we want to do is make a sample specific database. So the process goes a little bit like this, where we predict open reading frames from the PacBio derived transcripts, performing an in silico protein sequence generation. We compare the protein isoforms to the protein isoforms in the reference database, and I hope this image to your right at the top looks very similar to the process that we were undergoing when we were doing the transcript comparison. We can compare the protein the protein information that we have against what's already in the reference database to find known and novel protein isoforms. We can compile all this information into a HUVECs sample-specific protein isoform database. It's like a FASTA file that we can use for all of our downstream mass spectrometry analysis. So one of the thematic elements of the long-read proteomics pipeline that I think is very prominent is that we're able to find unique splicing patterns from the data that we find. Here I'm highlighting an example of PKM1, which is a critical marker for endothelial cells. At the top are the reference isoforms that have already been annotated within GenCode. In the bottom in the red box are the PacBio isoforms that we found through our own sequencing run. At the bottom, we have the sample identified peptides that we found as a result of mass spectrometry analysis. If I point your attention to where this variable splicing is happening as according to our PacBio isoforms, we see that this is happening within the cytoplasmic tail of PKM1. And PKM1 has many interaction partners as interacting within the cytoplasmic arm. So we can speculate and hypothesize that there could be different modulations in terms of downstream signaling events and cascades. So I want to then ask the question of how we can integrate both long-read sequencing data with this proteomics information to further enhance isoform resolution. Much like this first scenario where I can't identify from which isoform this peptide was contributed by, if I integrate the abundance information that's provided by my long-read data, I can be drawn to suggest that probably of these two isoforms within my population, that the bottommost isoform that's expressing at CPM, 75 CPM, is probably the most predominant species within my HUVEC population based off abundance alone. And then I can also integrate this further with the proteomics data. If I find a unique peptide for a particular isoform, and I also integrate this abundance information, I can be drawn to suggest that this 25 abundance CPM isoform within my HUVEC population is probably the most predominant species within my HUVEC cells. And to highlight an example where we were able to take all this information, integrate this, is for CH5, where we're also characterizing these unique splicing events. And if I draw your attention once again to the top with the reference isoforms in GenCode, and the bottom with the PacBio isoforms, at the bottom with the green with the sample identified peptides, all this information together was helping us to characterize this novel splicing event that we were finding within CH5, which is also a really critical member for endothelial cells in terms of their adhesion properties. So some future directions for this project is that we can look to maybe looking at the functional effects of isoforms to understand if particular isoforms are more stable than others. We are working on targeted assays that are being led by our senior scientist, Dr. Aaron Jeffrey, to improve coverage of endothelial relevant genes, but also any genes or isoforms that we're interested in terms of proteomics coverage. And lastly, within the Hershey lab at UVA, we're working on isoform validation assays where we can pick particular isoforms, manipulate them, and understand the effect of these specific isoforms on phenotype. And all these have implications to developmental changes and disease models. And lastly, this work would not be possible without the work of the Shankman lab and the Hershey lab, and it's such a pleasure to work with all these individuals. I want to thank my committee members and my funding sources, and thank you for your attention. Happy to take any questions.
All right, thank you, Madison. I just want to mention to the rest of the audience that if you're joining the next few sessions, you do have to exit this session and then join the next session. So I do want to leave probably like another minute for people to move to the other session. And I also want to, maybe I'll do this later, is I do have a, let's see. There's a lot of questions happening. I'm trying to figure out which ones. Okay, so maybe this is a question for you, Madison. Have you guys considered a site-like approach where they use oligo-tagged antibodies instead of mass spec to detect the proteins?
I don't think we've looked at that explicitly, but we are looking at different ways of like tagging and kind of really just identifying specific forms or isoforms at our protein level. Whatever those different there's a lot of different avenues I think we're looking at currently, but we haven't explored that.
Okay. And actually, I think a question I had also was you were showing the, using CPMs, counts per million abundances to kind of help resolve the ambiguities and, find new protein isoforms. But I think my other extended question with that is, are you able to look at, is that information possible, to see at a protein isoform abundance? And where I'm going is is there a correlation between the RNA level abundance with the protein abundance?
Yeah, we have looked at that correlation between the RNA and the proteins that we're finding, and we find a relatively strong correlation, but it's not a direct translation. We know that some protein isoforms were missing in terms of our mass spectrometry coverage, so it might be a term of the lack of a method there, but also we know that sometimes transcripts are not being produced, and there's many different ways that we can kind of interpret that or maybe validate that down the line, I think.
All right, thank you very much. For those of you who are joining the next few sessions, I will see you there.